Hello guys, I'm glad to see you, and today we will discuss the second topic on histology. I hope that you can see the presentation and that you can also hear my voice.
Hello, Professor. Everything is okay.
Okay, thank you. Then we will start. So, today we discuss structural and functional organization of a cell. We will discuss the ultrastructure. Ultrastructure means very, very tiny, small structures inside the cell. It means that we will discuss and we will see the pictures obtained with the help of electron microscope. So, we were previously discussing the types of microscopy. We have light and electron microscopy. So today we will discuss mainly electron microscopy pictures, and we will discuss the structure and function of organelles of the cell. So, as you remember, the cell is a part of a tissue. Tissue is composed of cells and extracellular matrix. And here in this picture you can see overall total description of cell organization. In this picture you can see all the organelles that can be found inside some cell. Different cells perform different functions, and it means that they will have different degree of development of different organelles. So, amount of every organelle developed in cytoplasm depends on the function and activity of a particular cell. The same in this picture. This picture shows everything that can be found inside the cell. So, overall, cell is composed of the cytolemma. It's the cell membrane covering the cell from outside. Here it is, the cytolemma. And inside we have the cytoplasm with organelles and some inclusions. And cytosol. Cytosol is the jelly-like watery internal component of the cell. And also the nucleus. Every living cell has the nucleus. So we start to discuss the organization of the cell from the cell membrane. Cell membrane consists of three components. The major component is the plasmalemma itself, or the membrane per se. It's the Latin meaning the self, or the plasmalemma. This is the lipid bilayer, which consists of two layers of phospholipids and proteins. The proteins compose about 50% of the membrane composition. So the membrane consists of phospholipids and proteins. Proteins can be inserted inside the membrane like this one, and such protein is called integral protein. Some proteins can be just attached to the membrane like this one, and such are called peripheral proteins. So this is the membrane or plasmalemma. The second component of the cell membrane is the glycocalyx. Glycocalyx is seen here. It consists of the carbohydrate chains. Carbohydrate means sugar. These molecules are sugar molecules attached to the proteins or lipids of the membrane. So if this carbohydrate chain is attached to the protein or to the phospholipid, it makes the glycocalyx. In this electronogram, which means the picture obtained from the electron microscope, you can see the top of the cell of the intestinal epithelium. it's the microvillus covering the cell surface. Microvillus is seen here. It's a small protrusion of the apical surface, top surface of the cell. So here you can see the top of this microvillus, and it is covered with bush-like network of carbohydrate chains of glycocalyx. So this is the second component. Third component of the membrane is the submembranous layer, or it is also called terminal web. It is the composition of the elements of cytoskeleton of the cell, which include microfilaments, the tiniest smallest ones, intermediate filaments. They are called intermediate because they are intermediate in their size between microfilaments, smallest, and microtubules, which are largest elements of cytoskeleton. So these three elements of cytoskeleton make the terminal web. Their function is to attach the membrane of the cell to the internal composition of the cell. The glycocalyx, as you can see here, it covers the cell from outside, and it has a lot of different functions. First, it acts as a physical barrier for a cell. It means that nothing can pass through the glycocalyx to enter the cell. So it's for protection of the cell. Next, it provides cell recognition and communication. When two cells come close together, they are attached to each other with the glycocalyx, and they can decide if they want to make the junctions, contacts, or not. So they can recognize themselves with the specific carbohydrate molecules in their glycocalyx. Next, glycocalyx provides the intercellular adhesion in the cells of one type. So if two cells recognize themselves as the cells of the same type, then they can decide to make the contacts, adhesion junctions between each other with also the help of glycocalyx. In some cases, the glycocalyx prevents cell-to-cell adhesion. For example, like in this picture, this is electronogram showing cross-section of the capillary. Capillary is the smallest blood vessel. So here is blood plasma inside the this capillary, and the wall of the capillary is made up with the cytoplasm of the cell called endothelial cell or endotheliocyte. So here is the nucleus of this endotheliocyte, and here it's flattened cytoplasm, which makes the wall of capillary. And the apical surface of the endotheliocyte, which is here, it faces the blood inside the lumen of the blood capillary. Its surface is covered with glycocalyx, which function is here to prevent adhesion of formed elements of blood, adhesion of blood cells to the surface of endothelium, and clotting of blood. So in the case of endothelial cells of blood vessels, glycocalyx is negatively charged, as the glycocalyx covering all the cells of blood. So both are negatively charged, so they cannot attach to each other, and it prevents adhesion to cells of blood to walls of blood capillaries. And finally, in some cases, the glycocalyx provides the digestion process, like it is here in the example of the intestinal epithelium cells. So here you can see three cells of epithelium of the intestine. Here is the nucleus of one of them, second and third. first and third are covered with microvilli, which are shown here on higher magnification, and microvilli are covered with glycocalyx. It's the same as in this picture, and in this case some of the molecules of the glycocalyx are not carbohydrate chains, but the protein molecules, and these proteins are enzymes which help to break down the dimers of food, for example dipeptides, into monomers such as amino acids. So this is the process of digestion, breaking down of the larger molecules to smaller ones, and these smaller molecules, amino acids, then can pass through the membrane into the cytoplasm of the cell. So in the case of the cell of the intestinal epithelium, glycocalyx also provides the near membrane digestion. Here you can see the functions of some cell membrane proteins. So these are integral proteins. It means that they are inserted inside the bilipid layer of membrane. Here actually you can see the parts of two cells. One of them is here, second is here, and some extracellular matrix between them. So what can we see here? One of the functions of the membrane proteins is adhesion of the cell to extracellular matrix. As you can see here, the integral protein is attached to the glycoprotein or adhesion protein of extracellular matrix, and this glycoprotein binds the cell surface to the collagen fiber here, and also to the proteoglycan chain with the hyaluronic acid inside it, in its core, etc. So one of the functions of such protein is adhesion of cell to extracellular matrix components. Second function is signal transmission. In this case, this protein is not just a protein, it's the receptor. And the ligand for a receptor, the signal molecule, can attach to this receptor, and it will cause some function of the cell, change of some function. The next function of membrane proteins is transport of molecules. In this case, this protein is not just a protein, it's the protein channel, ion channel for some ions. These balls are some ions that can pass through this ion channel from outside into the cell if this ion channel is opened. Ion channels can be opened or closed, and they allow the ion molecules to pass along the concentration gradient. In this case, this is also the transport protein, but it's not the channel, it's the pump. It's the ion pump that can pump ions against the concentration gradient with the help of ATP molecules. So this protein will be called ATPase, or ion pump. ATPase means that it breaks down ATP to ADP and phosphorus, and this causes release of energy, and this energy makes it possible to pump the ion through this ion pump against the gradation, the concentration gradient from the cell outside or backwards in different cases. Next function is the formation of intercellular contacts, junctions between the cells. These proteins are specific proteins. They are called occludins and claudins, specific names for such proteins that can attach together to adjacent cells. Here you can see the function of recognition of one cell by another cell. It means that such integral proteins have the carbohydrate, sugar, chain attached to their surface, and in the case when the carbohydrate chain of the glycoprotein of cell 1 is recognized by the cell 2, then these two cells can be attached together to make the junction. And here you can see another function, enzymatic activity. In this case, this is also not the just some protein, it's the enzyme protein which is inserted inside the cell membrane. In this case, it performs its enzymatic activity inside the cytoplasm of the cell. In the case of the enzymes in glycocalyx covering the microvilli of the intestinal epithelium cells, they perform their enzymatic activity outside of the cell. So it can be both like this or that. Next, we will discuss the mechanism of transport across the cell membrane of some substances and molecules. So the cell is exchanging with extracellular matrix with different molecules all the time. So these molecules should somehow pass through the membrane inside the cell or outside of the cell. And it can be performed with via different ways. So here you can see three types of the transportation across the cell membrane without consumption of cytolemal material. So here you can see that no vesicles are formed and the molecules just pass through the cell membrane. So here is the cell membrane, plasma lemma, extracellular matrix is here, and cytoplasm is here. So here you can see three types of such transportation mechanism. First, passive diffusion. This is for some very small molecules such as water molecule or gases or ions. They can pass just between the molecules forming the plasma lemma along the concentration gradient from higher to lower concentration. This is just passive diffusion. Next one, facilitated diffusion. This is for transportation of ions, glucose, amino acids, also along the concentration gradient, but with the help of some specific integrating proteins, transporting proteins, but without help of ATP molecule. And third one is the active transport of ions, and also in some cases of glucose. Active means two things. First, that it goes against the concentration gradient from lower to higher concentration of ions. And second thing, that it is performed with the help of ATP molecules. So this is the ATPase, the ion pump, sodium-potassium pump in this particular case. So it pumps the sodium ions in one direction and potassium ions in the opposite direction with the help of ATP molecule against the concentration gradient. Next, here you can see the types of the transport of some molecules with the consumption of cytolemma material, and during this the vesicles are formed, like you see here. And in this table you can see three types of endocytosis, and on the next slide, exocytosis. Endo means inside, so these are three ways of the entering of molecules into the cytoplasm of the cell from the outside. And Exo means outside, so here we have 3 ways of going out of molecules from the cell outside. So, endocytosis, 3 ways: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is for eating large particles. So, example of such process is the function of macrophage. Macrophages can eat large particles like amoeba cells can eat the particles of food. So here some large particle is eaten by the cell, and the vesicle containing this particle goes into the cytoplasm of the cell. Pinocytosis is when the particles are small or liquid. So just the pinocytotic vesicle is formed. And receptor-mediated endocytosis resembles the pinocytosis, but here the particles of the molecules are attached first to specific receptors, and then the vesicle is formed which contains these receptors attached to the membrane and the molecules attached to those receptors. This is more specific as the pinocytosis is. Exocytosis, also the vesicles are formed here. 3 types we have also here. First, secretion. It's when the cell synthesizes something. For example, if the cell is the cell of some gland and it synthesizes some substances, for example enzymes, which should go out from the cell. So first the vesicles are formed which contain those enzymes, then the vesicles go towards the surface of the cell, then they fuse with membrane and release these enzymes outside. This is the secretion. Excretion is for removal of wastes of the cell, something bad that should be removed. Recreation is when substances are not altered, not changed during the process of their transportation from one side of the cell through its cytoplasm to other side. So in this case the molecules just go through the cytoplasm of the cell with no change. This is for example for endothelial cell. So here can be, for example, the internal surface of the endothelium cell. So here can be blood, and here the tissue. So the function of the blood capillary is to pass the molecules from blood to tissues. So these molecules are not changed when they go through the cytoplasm of endothelium cell from blood to tissue, and the same from tissue to blood for some wastes, for example. Next we start to discuss the organelles. We have a lot of different organelles inside the cell, and they are divided into 2 major classes: membranous and non-membranous. Membranous are composed of membrane and some internal components. They are mitochondria with 2 membranes, lysosomes, peroxisomes, endoplasmic reticulum, smooth and rough, and Golgi apparatus. Those consist of 1 membrane layer. Non-membranous have no membranes in their structure, and they are ribosomes, 3 types of cytoskeleton elements: microfilaments, intermediate filaments, microtubules, and centrioles. They are for mechanical support, contraction, and some other different function. Also, organelles are divided depending upon their functions. So one of the functions is mechanical support and contraction. It's for elements of cytoskeleton. Also the function of catabolism, which means breaking down of some molecules. So this is performed by mitochondria, lysosomes, peroxisomes, and in some cases smooth endoplasmic reticulum. And anabolic system means for synthesis of some molecules. So these organelles can synthesize some molecules. So we start to discuss organelles with endoplasmic reticulum. Endoplasmic reticulum is subdivided into two subtypes: rough or granular endoplasmic reticulum, RER for short, rough endoplasmic reticulum, and smooth or agranular, SER, smooth endoplasmic reticulum for short. And in some cases transitional, it is something in between rough and smooth one. So why the granular is called so granular or rough one? Because it consists of cisterns, these elongated structures which are seen here in this electronogram, covered with these black dots. The surface of cistern is covered with black dots. These dots are actually ribosomes. Every black dot is one ribosome. They are attached to the surface of the cistern of rough endoplasmic reticulum, making it rough. Smooth endoplasmic reticulum, which is seen here, also has cisterns, but no attached ribosome, so it is smooth. Also inside the cell we can find not attached ribosomes. They can be single, like these 1, 2, 3 single dots. They are single ribosomes. They are not active. They are just waiting for some informational RNA to start their function. Their function is the synthesis of proteins. And when the informational RNA comes from nucleus, then the ribosomes attach to it and they start to produce protein molecules. So here you can see the process of producing protein molecule. And in such case, the ribosomes are not single and they are seen like small spirals, like you see here, spiral or circle groups. And such groups of active ribosomes are called polysomes or polyribosomes. Poly means many ribosomes in group. So these are actively synthesizing ribosomes. Their function is to produce proteins for the needs of the cell itself. And such proteins produced by free polysomes stay inside the cytoplasm of the cell. So such proteins can be, for example, the proteins of which elements of cytoskeleton compose of. For example, the tubulin proteins, which make the microtubules, elements of cytoskeleton. So these are produced by polysomes. If the ribosomes are attached to the surface of rough endoplasmic reticulum, like here, they also produce proteins, but in this case, these proteins will be located inside the cistern of the endoplasmic reticulum. And these will be some different proteins. So these will be the membrane, lysosomal and secretory proteins, and also here in rough endoplasmic reticulum inside it, the initial modifications and post-translational changes of protein molecules happen. So both polysomes and rough endoplasmic reticulum make proteins, but they make different proteins. Polysome make cytoplasmic proteins for the cell itself, and rough endoplasmic reticulum makes the proteins for membranes. It makes proteins for lysosomes, which are for breaking down the molecules inside the cell, and the proteins for export, which means the proteins which should go out from the cells. Here you can see the process of protein synthesis on the granular or rough endoplasmic reticulum. So this is the matrix informational RNA upon which ribosomal subunits are attached. Then these ribosomes attach to the surface of the rough endoplasmic reticulum membrane, and then they start to produce protein which goes inside the internal space of the rough endoplasmic reticulum here. So in this case, the protein is now inside this rough endoplasmic reticulum. Then this protein, polypeptide, is chemically changed, and then it can go inside the vesicle that will be detached from the rough endoplasmic reticulum next to Golgi complex. Here you can see the differences between rough and smooth endoplasmic reticulum. This is how they look like in 3D-dimensional picture. And if we make the cut, the section, this is how they look like in 2-dimensional pictures. So rough endoplasmic reticulum has elongated structures, cisterns, covered with the ribosomes. And this is how it really looks like in electronogram pictures. So these are cisterns covered with small dots, ribosomes. Rough endoplasmic reticulum. Smooth endoplasmic reticulum is composed of tubules, and these tubules go in different directions, and when they are cut, this is what we see. We see different shape vesicles and elongated cisterns, and this is how it really looks like in electronograms. So these are small vesicles, small tubules, small cisterns of different shape. This is actually the smooth endoplasmic reticulum, and no ribosomes are attached to the membrane. Here you can see some electronograms. Inside them you can differentiate rough endoplasmic reticulum. Here it consists of many elongated cisterns covered with very small dots of ribosomes. Also here rough endoplasmic reticulum, and here also another one. Another example of rough endoplasmic reticulum, and here rough and smooth endoplasmic reticulum. Smooth endoplasmic reticulum consists of different vesicles, tubules, different shape cisterns. Another example of it. And the functions of smooth endoplasmic reticulum include synthesis of lipids, including the membrane lipids. So also the membranes of smooth endoplasmic reticulum contain lipids produced by this organelle itself. Phospholipids of every membrane are produced in smooth endoplasmic reticulum. Next, glycogen synthesis, not in every cell, but in some cells, for example, the cells of liver, which synthesize glycogen and store it. Glycogen looks like dark inclusions like this. Next, cholesterol synthesis. Cholesterol is the lipid also included in cell membrane, so cholesterol is good. It is for membrane stability. Next function, detoxification of endogenous and exogenous substances. It means that some toxins are transformed into some non-toxic molecules inside the smooth endoplasmic reticulum. And finally, deposition of calcium ions. So calcium ions are stored also inside the smooth endoplasmic reticulum. In these pictures, you can see smooth endoplasmic reticulum here. Also here, different shapes of vesicles and tubules. Also in this part of the picture. And in this picture, you can see these small vesicles. They are vesicles of smooth endoplasmic reticulum. And these patches of dark dots, they are actually granules of glycogen. So this is the part of cytoplasm of some cell which function is to produce and store glycogen. Next, Golgi apparatus. Golgi apparatus consists of many plate-looking cisterns attached together, and it has the cis and trans part. Cis part looks towards the cell nucleus, so cell nucleus is not seen in this picture, but it should be here. And this is the trans part of the Golgi apparatus. It always faces the surface of the cell, so surface of the cell should be here. The cis portion of Golgi apparatus is exchanging with vesicles with smooth and rough endoplasmic reticulum. So everything which is synthesized inside rough and smooth endoplasmic reticulum then goes inside vesicles towards the Golgi apparatus, where these synthesized molecules are next changed somehow. For example, glycosylation, attachment of carbohydrate residues, and something else can happen to these molecules. And while this happens, the vesicles go through the Golgi complex, and finally these modified molecules are in trans portion of the Golgi apparatus, and here the sorting of molecules happens, and Golgi apparatus decides whether this particular molecule should go outside from the cell via secretion or excretion, or this particular substance is the enzyme that should stay inside lysosome, or something else. So, functions of Golgi apparatus include sorting of proteins and other molecules that are synthesized inside rough and smooth endoplasmic reticulum and go to Golgi apparatus. Next, processing of molecules. It means that these molecules are chemically changed somehow. Next, synthesis of polysaccharides and glycoproteins of glycocalyx and mucus. So glycocalyx is covering the surface from outside. It consists of carbohydrate chains. So these carbohydrate chains are synthesized inside Golgi apparatus itself, and mucus also. Next, condensation of a secretory product. It means that these molecules become more concentrated, water is removed, and the solute becomes more concentrated. Next, membrane packaging of secretory products. It means that these molecules are covered with membrane of Golgi apparatus cistern and become vesicle with some content. And finally, formation of secretory, hydrolase, and membrane vesicles. So different vesicles detach from the trans pole or portion of Golgi apparatus. So this is the feature of trans pole of Golgi apparatus. Here you can see it. and near it you can see a lot of vesicles which are detaching from the surface of Golgi, and then these vesicles will go this way. Some examples of Golgi complex. So this is Golgi apparatus on the left picture. Here you can see the portion of the cell nucleus. So this is the cis pole, cis portion, and this is the trans portion from which some vesicles are detaching. Another example, so here will be cis pole, this will be trans pole from which vesicles are detaching and go some this way. So all the membrane organelles inside the cell are actually interconnected with each other. So like you see here, the rough or granular endoplasmic reticulum is continuous actually with the membrane of cell nucleus, and rough endoplasmic reticulum is continuous with smooth one, not shown here. Next, everything which is synthesized here goes inside vesicles to Golgi complex to its cis portion, then is detached from the trans portion of Golgi and goes in different direction depending on the nature of these molecules. So here you can see the role of Golgi apparatus in the processes of exocytosis and endocytosis. So exocytosis means release of some molecules outside from the cell. So something is produced in rough endoplasmic reticulum, some enzyme, for example, it goes through Golgi complex, modified here, covered with membrane, and this secretory granule goes to the cell membrane, where it fuses with the membrane and releases the molecules outside. Next, what can also happen in the case of endocytosis, phagocytosis. In this example, you can see that the cell is phagocytosing the bacterium. This is some bacterium which is eaten by the cell. So during this process, the membranous vesicle is formed which covers the bacterium inside it. This vesicle goes to the towards the Golgi complex, and from Golgi complex the vesicle containing enzymes is detached and it goes towards this phagocytic vacuole, which is called phagosome, vesicle containing bacteria. Next, the vesicle with enzyme fuses with this vesicle containing bacterium, and the enzymes start to digest bacterium. And this vesicle containing enzymes and what they are digesting, in our case bacterium, this will be called phagolysosome or secondary lysosome. Phago because it's from phagocytosis, lysosome because it's from lysosome, the vesicle containing enzymes. So phagolysosome. Here enzymes in acid environment digest bacterium. Every good substances, for example sugars and amino acids, go outside from this vesicle to cytoplasm, and residual body which are non-digested remnants of this bacterium go outside from the cell via excretion process. In this case, some damaged self organelle, for example damaged mitochondrium, which should be replaced with a new one, so the old one should be broken down. So this mitochondrium will be enclosed with the membranous vesicle, with which lysosome will fuse to make the autophagic vacuole. Auto means itself, so it means that the cell breaks down its self organelle, not something from outside, but its own damaged organelles. Phagic means the same as for phagocytosis. So this broken, damaged mitochondrium will be digested with the enzymes from lysosome. And next, the same process, everything which can be utilized by the cell itself will go outside, some sugars, lipids and amino acids, and what cannot be broken down makes the residual body, which evacuates the non-digested particles outside from the cell. So in these processes, lysosomes play the major role. So functions of lysosomes include breaking down of polymers into monomers. It means like proteins to amino acids. And next, diffusion of monomers into cytoplasm. So here you can see the secondary lysosome, like it is here, phagolysosome, because it's the vesicle with one membrane layer containing some content with different colors, both darker and lighter colors. It means that here previously was something which is now partly digested, and different parts are digested with different speed, so you can see here different colors. So this is the secondary lysosome. So endosomes functions. Endosome is any vesicle with some content inside the cytoplasm of the cell. So these endosomes can transport molecules from cell surface to lysosomes, like the phagocytotic vacuole here. Also, they perform pre-lysosomal stage of destruction of substances, receptor ligand complexes, denaturation of proteins, etc. So in some cases, the digestion of some internalized particle can start before it fuses with lysosome in some cases. Next, peroxisome is the small organelle, also with one layer of membrane covering it, and its content has this very funny looking so-called crystalloid or nucleoid. Actually, it's the mixture of specific enzymes of peroxisome. They start to look like this as a result of the histological preparation of tissue for electron microscopy. So crystalloid contains enzymes. Enzymes are for performing functions of peroxisomes. So functions of peroxisomes include protection of cells from hydrogen peroxide. It means that peroxisome contains enzymes that break down hydrogen peroxide, H2O2. This is the chemical formula of hydrogen peroxide. It is harmful for any cell. So peroxisome breaks H2O2 and protects the cell. Next, peroxisome neutralizes some certain substances, for example, ethanol and other alcohols. Also, peroxisome enzymes catalyze the breakdown of fatty acids and some other catabolic and anabolic reactions. Catabolic are for breaking down, and anabolic means synthesis of something. So here are some examples of electronograms that can be shown to you, and you should be able to find the organelles here and describe their functions and how they look like. So what do we see here? Among some mitochondria, which we see next, here some vesicles of different shape. They are actually parts of smooth endoplasmic reticulum. These dots are ribosomes. They are actually polyribosomes, some groups of ribosomes. And these are 4 lysosomes. This one is equally grayish in looking, so this should be the primary lysosome just containing enzymes, and these have content of different degree of black and white, so these are secondary lysosomes. They are already digesting something. This should be peroxisome with this funny looking crystalloids inside it. This is the autophagosome. It means the vesicle containing some organelle that will be digested next with the help of lysosome. So this one is the mitochondrium with cristae inside it, and this is the autophagic vesicle. Also here outside you can see some vesicles of smooth endoplasmic reticulum of different shape. These are granules of glycogen and also some ribosomes. Glycogen looks very similar to ribosome. It is not very simple to distinguish between them. And also in last one you can see the secondary lysosome with content of different colors. And also some mitochondria, polysomes, polyribosomes not attached to anything, and smooth endoplasmic reticulum vesicles. Next, mitochondria. Mitochondrion is one mitochondria, and mitochondria are actually plural name for these organelles. So mitochondria contain 2 membranes, the outer membrane and internal membrane, and this internal inner membrane makes folds, and these folds are called cristae. Cristae is the plural from crista, one crista, many cristae. And these cristae are separated with matrix, some content which is between the cristae. Inside these cristae, the ATP synthase is inserted. These mushroom-looking molecules are the molecules which produce ATP. So it makes us easy to see that the major function of mitochondria is producing ATP for the cell. Also, we have 2 types of mitochondria. First, in all the cells we have the mitochondria with lamellar cristae. Lamellar means some flattened and elongated structure, like it is shown here. So this is lamella. And second type with tubular vesicular cristae. In this case, the cristae are not like longitudinal folds. They instead look like elongated tubules or vesicles. This is how these types of mitochondria really look like in electronograms. So this one is mitochondrion with lamellar cristae. Here they are elongated structures, and in this picture you can see the mitochondria with tubular vesicular cristae, and you can see here these vesicle looking cristae. The feature of this second type is that it can be present only in those cells which produce steroid hormones, such as the cells which produce, for example, testosterone and estrogen hormones, sex hormones, which are all steroids in their chemical nature. So the cells which produce these hormones have these very specific mitochondria with tubular vesicular cristae. And all the cells of our body contain this first type, mitochondrium with lamellar cristae. So functions of mitochondria include first, they provide the cell with energy obtained by oxidative phosphorylation. Oxidative phosphorylation means that organic molecules are oxidized, so they are breaking down with the help of oxidation process, and as a result of this process, the energy is released. And second step of this process, oxidative phosphorylation, includes phosphorylation of ADP. It means that ADP plus phosphorus, as a result, we have ATP. So, as a result, the mitochondria produce ATP. Second, mitochondria participate in the synthesis of some substances such as nucleic acids and some proteins, fatty acid oxidation. Also, these specific are producing steroids. Also, mitochondria participate in process of apoptosis. Apoptosis means the programmed cell death. Many cells should grow, age, and die in the end, and then they are replaced by the newly produced cells. And this programmed death of the cell is called apoptosis, and it needs a lot of energy. So it is not just breaking down of the cell and all of its content, but it's programmed, very complex process which needs a lot of ATP molecules. So mitochondria give ATP and also a lot of enzymes which take part in this complex process. And also mitochondria involved in maintaining of calcium homeostasis. And usually we can see these inclusions of calcium. These dark looking dots are actually the calcium crystals inside mitochondria. So, some other examples of electronograms. In the left upper one, in the upper portion of it, you can see some rough endoplasmic reticulum parts. In lower part here, you can see Golgi complex with this cis portion facing nucleus. This is the part of nucleus, and trans portion with vesicles detaching from it. These small dots are free non-active ribosomes. Here in the right upper picture you can see the top surface of some cell, because you can see here these small protrusions, microvilli, which can be found only in the apical or top surface of the cell, and some portion of its cytoplasm, with a lot of smooth endoplasmic reticulum, which looks like a lot of vesicles and tubules of different shapes. And these are mitochondria. Mitochondria have two membranes, and inner one makes these folds cristae. In lower left picture you can see smooth and rough endoplasmic reticulum and also some mitochondria. Mitochondria, regular one with lamellar cristae. And here in the lower right picture you can see a lot of rough endoplasmic reticulum and a portion of the nucleus of the cell. In these two pictures, in the upper one you can see polyribosomes, spiral looking and circle looking. So they are active free ribosomes making cytoplasmic proteins for a cell itself. And in lower picture you can see the mitochondrium with lamellar cristae, with DNA and calcium content, and some endoplasmic reticulum. Here, in the upper picture, mitochondrium with lamellar cristae here, and in left part insertion, these are mitochondria with tubular vesicular cristae. In upper right picture, you can see mitochondrium with lamellar cristae and rough endoplasmic reticulum, and lysosomes, these vesicles with dark content. In lower left picture, you can see mitochondria with lamellar cristae, smooth endoplasmic reticulum, vesicle looking, and granules of glycogen produced by smooth endoplasmic reticulum. In lower right picture, you can see dividing mitochondria. It means that new mitochondria are produced by dividing of some mitochondrium inside the cell. Old mitochondria are utilized with the autophagocytosis with the help of lysosomes, so they are broken down, and new ones are produced as a result of division of mitochondria. Next, we go to the cytoskeleton of the cell. Cytoskeleton is like bones to our body, so their major function is to produce the internal scaffold, internal strength of the cell, to maintain its shape. And we have three components of cytoskeleton: microtubules, microfilaments, and intermediate filaments. They are called so depending on their diameter of these elongated structures. So microtubules are the thickest. Their diameter is 25 nanometers. Microfilaments are the smallest. Their diameter is 7 nanometers. And intermediate ones are intermediate. In their size, they are 8 to 12 nanometers. So all of them are present in every cell. Cells are shown in the upper panel. All of them, microtubules, microfilaments, intermediate filaments, they consist of proteins which make monomers. Every protein, like this alpha and beta tubulin for microtubule, or G-actin for microfilament, or monomer of intermediate filament, they are monomers, and they are combined together to form larger structure, polymer. So microtubules, this is how microtubule looks like in elongated look, and this is how it looks like in cross section. If we cut it like this is how it will look like. And its diameter, it is 25 nanometers, and it consists of two types of proteins, alpha tubulin and beta tubulin, which make the dimer, two molecules combined together. Microtubules are seen here in the lower panel. These are two electronograms. In the first one, you can see their longitudinal section, these ones, and in second one you can see them in cross section, so they look like small, very tiny circles. The monomer proteins, tubulins, are produced by free polyribosomes inside the cytoplasm of the cell. The functions of microtubules include formation of internal scaffold, maintenance of shape. Also, they provide intercellular transport of organelles and vesicles. It means that every mitochondrium, for example, which is here, it is attached to this microtubule, as you can see here. Mitochondria can move along the processes of the cell. For example, if a cell has processes, with the help of some specific proteins which move these organelles along microtubules. So microtubules are kind of railways inside the cells for movement of all the vesicles and organelles. So nothing just flows inside the cytoplasm of the cell, but instead it everything goes along the railways of microtubules. Next, formation of spindle apparatus during cell division, and participation of movement inside specific structures for movement, such as cilia, flagella. Flagellum is the tail of spermatozoon, as you know. So spermatozoa in male body, they can move because they have tail, and inside their tail they have specific microtubules, and these microtubules help the tails of spermatozoa to move. Next, microfilaments. Microfilaments also contain monomers, monomer proteins. Mono means one, poly means many. So monomers are combined together to form polymer molecules. Here in this picture you can see the example of the microfilaments, which are characteristic for muscle cells, muscle tissues. They are actin and myosin filaments, which feature is that they can move along each other in different direction. For example, in this case, the actin filament will go this way, leftwards, and myosin will go rightwards. So they will go along each other, and it will cause contraction. Functions of microfilaments. So first, they participate in contraction, especially in contractile tissues such as muscle tissues. Next, they form the terminal web. As you remember, terminal web is the network of cytoskeleton elements just beneath the cell membrane of the cell. Terminal web is here. Next, they ensure cell motility. It means that because they are located just below the membrane of the cell, when the cell needs to make the pseudopodia, for example, some protrusions like amoeba or macrophage have to move, the formation of such pseudopodia is ensured by microfilaments. They are contractile, so they can help the cell membrane to change its shape, and so they ensure cell motility. Also, they participate in formation of outgrowths, microvilli, which are shown here, microvilli covered with glycocalyx. This is the cell of the intestine. Also pseudopodia for movement, etc. Also, they provide phagocytosis. It's also the same function, because it is everything about the changing of the shape of the cell membrane with the help of contractile microfilaments just below it. So during phagocytosis, the endocytotic vesicle will be formed. So microfilaments will cause invagination of the cell membrane. Also, they participate in formation of focal contacts and belt desmosomes. Focal contacts are the contacts of the cell with the extracellular matrix, temporal contacts, and belt desmosomes or adherent junctions are the mechanical junctions between two adjacent cells. So here you can see the border between two cells, and one of specific junctions between these cells include these mustache-looking fibers of microfilaments. And also microfilaments provide formation of constriction during cytotomy, during cell division. Intermediate filaments also are made up of monomers, which first form dimers. Mono means one, di means two. Next, dimers form tetramers. Tetra means four, and tetramers make protofilaments and finally intermediate filaments. This is how they are fusing together to finally make the intermediate filaments. A feature of the intermediate filaments is that they are made up of the proteins which are tissue specific. We actually have 4 types of tissues: epithelial, connective or internal environment, muscle, and neural tissue. And the cells in this class of tissue have the specific proteins. So the cells of epithelial tissue will have intermediate filaments which consist of keratin protein. In connective tissue of vimentin protein, in muscle tissue of desmin, and in neural tissue where we have neurons and glial cells, we will have neurofilament proteins in neurons and glial fibrillar acid protein in glial cells. And this feature helps oncologists, the doctors whose specialization is oncology, cancers. They can look at the section of some tumor and say which tissue makes this tumor, and it will help to give some specific medicine to cure this particular cancer. So it is very important. Functions of intermediate filaments: first, and the major one, maintaining of the cell shape. Next, distribution of organelles within the cell, and distribution of deformation forces under mechanical load on cells. It means that when we push our skin, for example, it will not lead to breaking down of the cells, because the intermediate filaments, which go from one side of the cell to other one, and then in next cell the same, they kind of distribute this force and protect the cell. Next, participation of formation of desmosomes and hemidesmosomes. These are specific junctions. Desmosome is the specific junction between the cells. Here is one cell, and here should be second cell, with which it is attached with the help of desmosome. And hemidesmosome is the specific junction of the cell with the basement membrane upon which the cell is lying. It will be discussed next time. And in epithelium tissue, they participate in formation. Formation of the stratum corneum of the keratin. This is how the intermediate filaments look like if we use the luminescent microscopy, not our general light one. So the microfilaments are located mostly at the periphery of the cell, just below the cell membrane, and microtubules and intermediate filaments fill the cytoplasm. Here you can see some cell. It has junctions with adjacent cells and also with the extracellular matrix. These are specific junctions. In all of them, one of its portion is some element of cytoskeleton. So the cells are filled with these cytoskeleton elements, which help them to make the internal scaffold, because all of them are interconnected together, and the organelles are connected to the cytoskeleton elements, and also these cytoskeleton elements are connected to the junctions. Next, you have this specific task in your album, which is the description of the ultrastructure of the cell depending on its specific function. As I said previously, different cells perform different functions, and it means that they will have different degree of development of different organelles. It means that every cell will have nucleus, rough and smooth endoplasmic reticulum, Golgi apparatus, cytoskeleton, etc., but different amounts of these organelles depending on their functions. So, first, the cell which is specialized on producing of proteins. We will discuss the glandular cells. Glandular cells means the cells of glands. Glands are organs which produce some substances. So they are formed with glandular cells which produce and release outside some substances. So first, protein producing cell. It will have a very well developed rough endoplasmic reticulum because it's for production of proteins for export. Here in the left picture you can see it labeled with number 1, a lot of rough endoplasmic reticulum. In right picture you can see here many cells. One of them is here. Here is its border, a border of second cell is here, and some other cells. We look just at this one cell. So number 1, everything here around the cell nucleus is here, so everything outside of the nucleus here is actually rough endoplasmic reticulum. Second, Golgi complex, because it's for making secretory vesicles or secretory granules. Golgi complex is located between rough endoplasmic reticulum and granules. So it should be located here. Usually it's located above the cell nucleus. Cell nucleus is here, granules number 3 are here, those dark electron dense circles, and Golgi complex should be here. In this picture it is located here. And secretory granules or secretory vesicles, they are located at the top portion of the cell, because when it will be needed, they will be released from this top surface of the cell outside of the cell. So in the left picture you can see a portion of the serocyte, the part of its nucleus and part of its cytoplasm. A serocyte is a cell of salivary gland. Salivary glands produce saliva, which fills fluid which fills our mouth. And saliva contains also some proteins, for example antibacterial proteins, and these proteins are produced by serocytes. And in the right picture you can see the acinocytes. One of them is here, the cells of pancreas. They produce enzymes of pancreas, and these are enzymes for digestion. In light microscopy, how this cell will look like. Rough endoplasmic reticulum is stained always basophilic because it contains ribosomes, and ribosomes contain RNA, and they will be stained basophilic as DNA in cell nucleus. So if a cell has a lot of rough endoplasmic reticulum, it will have basophilic cytoplasm. So here you can see a group of cells producing pancreatic enzymes, and here is their basal portion where a lot of rough endoplasmic reticulum is seen around the cell nucleus. So this portion is more violet, bluish violet, more basophilic because of rough endoplasmic reticulum. Golgi complex is not stained with our dyes, and secretory granules are usually stained oxyphilic. It means that they will be stained with eosin, and they can be seen in this portion. So here you can see these secretory granules. So in left picture, this is light microscopy image, and in right picture, the image from the electron microscope. Electron microscope is black and white, and light microscope image stained with hematoxylin and eosin is of two colors. Next, the lipid-producing cell will have a lot of smooth endoplasmic reticulum, or histologists say that they have well-developed smooth endoplasmic reticulum, because smooth endoplasmic reticulum is the organelle where lipids are synthesized. Next, Golgi complex for packaging of these lipids, and finally secretory vesicles, secretory granules. So this is the sebocyte, a cell of the sebaceous gland. Sebaceous glands are located everywhere in our skin, and they produce sebum, which is mixture of lipids, which make our skin fatty looking, and also which make our hair, which grow from skin, also fatty looking. So this cell has nucleus, it is here, cytoplasm. In cytoplasm, a lot of secretory vesicles, number 3, filled with lipids. And in cytoplasm, here, we will have a lot of smooth endoplasmic reticulum. In this picture, left part, we have smooth endoplasmic reticulum. And in left upper picture, we have the picture of sebaceous gland, actually, with the cells, here they are, which during their life cycle become filled with these vesicles with lipids, and then they finally release their lipids upon the surface of the skin. How it will look like in our light microscopy in stained sections. So this is the sebaceous gland. This is how it really looks like in our microscopy. So here we can see the cells with cell nucleus inside them, and you can see here that every cell is filled with a lot of non-stained vesicles. These vesicles contain lipids, they are not stained. Smooth endoplasmic reticulum also is not stained, and Golgi complex also is not stained. Next, steroid-producing cells. Steroid is the class of lipids, actually, and the example of such cell which produces steroid is the secretory cell of adrenal gland cortex. Adrenal gland, its cortex part, produces steroid hormones. For example, male sex hormone, testosterone. So here you can see the portion of such cell. A part of its nucleus is in the left part, and a portion of its cytoplasm, inside which we have this very specific mitochondria with tubular vesicular cristae, number 1, and also these rounded structures with vesicles and tubules inside them. So these are vesicular and tubular cristae. Between the mitochondria inside cytoplasm, we can see vesicles and tubules of different shape. These are smooth endoplasmic reticulum, which is for lipid production, and these grayish are lipid droplets. They are actually the inclusions with the So the cytoplasm between producing cell. It makes mucus. Mucus is some jelly-like fluid which fills your nose and mouth. Mucus actually contains water and mucins. Mucin is the large molecule which is actually heavily glycosylated protein. It means that mucin molecule contains two parts, protein part and sugar, carbohydrate part. And now you know that such cell should have two well-developed organelles. One for production of protein molecule, it will be rough endoplasmic reticulum, and second portion of the molecule of mucin is carbohydrate, and we know that Golgi complex is for production of carbohydrates of mucus, and also secretory granules or vesicles. So example of such mucus producing cell is the so-called goblet cell, which is shown in both pictures here. Goblet is the name of this glass of very specific shape. It has its larger portion where some fluid is stored, and a thin portion like here, and this describes the shape of the goblet cell. So this is the goblet cell. In its lower thin part we have the nucleus, triangular in shape, and some amount of cytoplasm. And in this cytoplasm we will have rough endoplasmic reticulum, the same in this picture. In lower part, nucleus, it is surrounded with cytoplasm filled with very well developed rough endoplasmic reticulum. Number 1. Golgi complex is usually located above the cell nucleus here. It will be here. And number 3, secretory granules, vesicles. So most of the volume of this cell is actually filled with the vesicles containing mucus. And when it is needed, the cell receives signal and it will release these vesicles containing mucus outside. In light microscopy, how the cell will look like? Rough endoplasmic reticulum is stained basophilic. So, electronogram, light microscopy stained with hematoxylin eosin. This is goblet cell. This is its nucleus. It is surrounded with small amount of cytoplasm, and this cytoplasm is stained basophilic. Golgi complex is not stained, it's above the nucleus here. And secretory granules containing mucus are also not stained. So these cells will look like white bubbles in histological sections. So this is the section of the intestinal villi covered with epithelium, and inside this epithelium we have these goblet cells which look like whitish bubbles, because these white bubbles are upper portion of the cytoplasm filled with not stained granules with mucus. The goblet cells are surrounded with the major cell type of the epithelium of intestine. Here they are, these cells with microvilli covered with glycocalyx for digestion. Next, cell producing glycogen. This is carbohydrate. Glycogen is the form of storage of carbohydrates in the human body, and glycogen is mainly stored inside the liver. Glycogen is produced by smooth endoplasmic reticulum. So in such cell we will see two major features: smooth endoplasmic reticulum and glycogen granules. So here is such cell, and in the left picture also. Here in the right picture you can see in the upper part a portion of the cell nucleus and the portion of its cytoplasm, inside which we see some mitochondria, rough endoplasmic reticulum, and these darker inclusions, dark black inclusions, they are glycogen granules. And between them, not very well seen, but somehow they are seen, vesicles of different size and shapes, they are smooth endoplasmic reticulum. The same in this picture, mitochondria, and number 1 is for smooth endoplasmic reticulum, vesicles of different shape and size, and number 2 for glycogen granules, dark inclusions. In light microscopy, in our general hematoxylin and eosin staining, the glycogen is not seen because it is not stained as the smooth endoplasmic reticulum is. But if we use specific staining, which is called PAS staining or PAS reaction, which is used in hospitals usually, we will see the glycogen granules which are red in color in this specific staining. So in light microscopy you can see here a lot of hepatocytes, liver cells with their cell nuclei, and reddish inclusions of glycogen. Also, the cells of liver store lipids. Lipids are not stained, they look like whitish circles inside the cytoplasm. Next, cell nucleus. Cell nucleus, shown in here. Its functions are that it stores genetic information in form of DNA. So DNA of the cell, most of it is inside the cell nucleus, but also some DNA is located inside mitochondria. Next, cell nucleus controls cell activities via genes expression. It means that all the cells of our body have the same number of genes. So every cell have the same genes, but different cells look different and they perform different functions. How it can be so if they have the same genes? It's the result of different expression of genes. Genes are portions of DNA encoding proteins, and these proteins can be enzymes which help to perform some functions. And in different cells, different genes are active and other genes are blocked. And in most of the cells, 99% of genes are blocked and only 1% of genes is active. And this 1% is different in different cells. For example, in neurons we have up to 10% of genes active, so neurons are very active cells. So this happens inside nucleus. And also inside the nucleus, initial step of protein synthesis happens. Protein synthesis includes transcription, which means that DNA is transcribed into informational RNA molecule, and this RNA molecule goes from nucleus then to cytoplasm, where ribosomes are attached to this RNA and start to produce protein, and this step will be called translation. So first step of protein synthesis is happening inside the nucleus. Cell nucleus has the membrane. It is actually double layered membrane, which is called nuclear envelope. So it has internal and external membrane, and as you can see here, external membrane of the nuclear envelope, outer nuclear membrane, is actually continuous with the granule or rough endoplasmic reticulum. It is also covered with ribosomes. Inside the nucleus itself, we have the karyoplasma, the internal content, inside which we have the chromatin. This is the DNA plus specific proteins in form of hetero and euchromatin, and also nucleolus. The internal content of nucleus is not fully separated from the cytoplasm of the cell, but we have some gates between the nucleus and the cytoplasm, which are called nuclear pores. Here we can see these nuclear pores. In this electronogram, you can see a part of the nucleus with the nuclear envelope consisting of internal and external membrane, and the nuclear pore, which is between the nucleus itself and cytoplasm. The DNA is packed inside the nucleus. While it gets packed, it is covering the specific proteins, histone proteins, and DNA, which contains all the genes of the cell, is differently packed. So different portions of DNA are packed in different degree of compactization. And as a result, we have some lightly looking areas, which are called euchromatin, and more dark looking areas, which are called heterochromatin. Heterochromatin areas are more densely packed DNA. So in nucleus, here is nucleolus, we are not discussing it now. In the nuclear plasma, we have euchromatin, which contains active DNA, and heterochromatin, more dark looking. It is non-active DNA. It is non-active because it is more densely packed. If the DNA is active, it should be not packed. It should be free to perform the transcription, the information from DNA to RNA during the protein synthesis. So the genes which are active in these particular cells will be in form of euchromatin, because they are ready to express these genes to proteins, and all other 99% of DNA will be in the form of heterochromatin. It is non-active DNA. So some cells will have more light looking nucleus with a lot of euchromatin, with a lot of active DNA, and some cells will have more small and dark and densely packed nucleus. So if a cell has a large and light nucleus with well-developed nucleolus inside it, it will give us information that euchromatin prevails in this cell, and it means that this cell is now actively producing proteins, and this cell is active. In this picture, actually, you can see the neuron cell, which is surrounded with glial cell. This was your picture from your first class. So neuron is very large cell. It has very large and light nucleus. It is light because of euchromatin, and it means that this cell is producing proteins now. But the glial cells have small dark nuclei. Here they are, small dark. It means that inside them heterochromatin prevails, which is non-active DNA, and it means that these cells are not so active in the function of producing proteins. So looking upon the nucleus of the cell, we can say if it is active or not. Nucleolus. Nucleolus function is production of ribosomes. So all the ribosomes of the cell are actually produced inside nucleolus. And nucleolus has three components. First, amorphous or nuclear organizer. It is more lightly looking area. It contains DNA with gens encoding ribosomal RNA. So these area have DNA with gens for making ribosomal RNA. Next, fibrillar component, more darkly looking area, contains newly synthesized ribosomal RNA. And granular component, which looks like small granules, dots, contains newly synthesized subunits of ribosomes. So the process of production of ribosomes include step 1, making ribosomal RNA from the nuclear organizer here, DNA, which encodes ribosomal RNA. And newly formed ribosomal RNA will form fibrillar component of nucleolus. And next, the proteins, which will go from cytoplasm to the nucleus, will help to produce subunit of ribosome. Ribosomal subunit contains ribosomal proteins and ribosomal RNA. And these subunits will make granular component of the nucleolus. So if we see the cell with well-developed cell nucleolus, it will tell us that the cell is actively producing ribosomes, and it is also the feature of the cell which is actively producing proteins, because ribosomes are for making proteins. Nuclear pore is not just the hole inside the nuclear envelope, inside the membrane of the nucleus, but it has very complex structure. It has the central granule in the middle portion, is here. Also, it has peripheral proteins in both outer and inner side, which are these rounded structures, peripheral portions, and fibrils, which are attached to these peripheral proteins. Inside the nucleus, the so-called nuclear lamina is attached to the internal surface of the nuclear membrane. Nuclear lamina, it consists of specific intermediate filaments, lamins, and it serves for attachment of euchromatin and heterochromatin to the nuclear envelope. The nuclear pores are for transporting of some molecules from cytoplasm to cell nucleus and backwards from nucleus to cytoplasm, and also for transportation of newly synthesized ribosomes from nucleolus, from cell nucleus towards the cell cytoplasm. And finally, we go to inclusions. We were discussing organelles, which are present always in any cells, but inclusions are temporary inclusions of molecules. So one of such inclusion can be the inclusion of glycogen, and you can see these inclusions stained in red color with specific staining, not hematoxylin eosin, but specific PAS reaction, periodic acid Schiff reaction. And hematoxylin was used to visualize cell nuclei. It's actually the liver of this funny creature, which is axolotl. At high magnification, you can see these cells hepatocytes. I'm showing the border of one such cell with its nucleus, and cytoplasm filled with red-colored glycogen granules. The same is here. The same section of the same liver of axolotl, but stained with osmic acid. Osmic acid, as you remember, is for staining lipids. And now glycogen granules are not seen, but instead we can see the lipid droplets. So these dark black and brownish looking droplets are actually lipid droplets inside the cytoplasm of hepatocytes. And in this specimen, more heavier staining, it means that this liver has higher amount of lipid droplets in hepatocytes. And also here we can see the pigment inclusions. Pigment, for example, melanin can be found in the melanocytes. The cells which are seen here, these are melanocytes. They are also present in our skin, which give the specific color to our skin and also our eyes and hair. These specific pigments in melanocytes. But in our preparation, we see the skin of a tadpole. So here you can see the tadpoles. They are baby frogs. So they will grow to frogs. And this is not stained preparation. No stain was used, but the cells are seen because they contain these granules of melanin. And fun fact is that the tadpoles can live in different lakes and pools, and in different environment conditions they will have different color of their skin. For example, if they live in some dark water pool, they will have darker skin to protect them from some other animals that can eat them. If they live in some lighter stained, in some light pool with a lot of sun lights, so then they will be not stained, also to be less visible for some animals that can eat them. And this is regulated by the amount of melanin granules in the melanocytes and the amount of melanocytes themselves. And the same, our skin can have a different degree of staining depending on our nationality and race, and also depending on the amount of sunlight which goes up to our skin. So we can go to the sea, and then our skin will grow darker because of larger amount of melanocytes. Here how they look like at high magnification. So the cell has its nucleus, cytoplasm, and processes filled with melanin granules. So this is all for today. If you have any questions, you can ask it now, or you can ask it using WhatsApp or anything else. So I will be glad to answer any of your questions. And also I will send this presentation to you.
Excuse me, Professor. I have a question that related to a previous lesson. I don't know how should we recognize the slice of the tissue. I know a little, but I think it's confused and I can't do.
Excuse me, once again, what didn't you understand in previous topic?
How should we recognize the slides of the tissue? For example, section, totally preparation.
Okay, I'll try to help you with this. So, types of microscopy. First of all, you should decide if you see the electron microscopy or light. Electron microscopy is in this picture and in this, and it is always in black and white, no color. If you see something with color, this is light microscopy. And next, here, types of histological preparations. You just have to, I think, learn these pictures because you are not familiar yet with these particular specimens, but maybe you have seen before the smear of blood. Maybe if you have some medical lessons before. So this is how the blood smear really looks like. Maybe you're familiar with red blood cells, which are look like this with lighter area in their center. So if you see them, you can say that these are red blood cells, cells of blood. So this histological preparation is the smear of blood with erythrocytes, red blood cells, and leukocytes. In this particular preparation, you can see cells with thin tails which grow from the cell, from the head. And this is how the spermatozoa look like. So if you see these spermatozoa with their tails, you can see that this is total preparation, because here the total cells are seen. They are not cut. And here section and whole mount, and I think that you have no other option but just to learn it.
Excuse me, Professor. For example, the cartilage of the ear is the section?
Yes, the cartilage is the section. Here is it. Yes, it is the section.
Okay, thank you, Professor.
And also, almost all the preparations are sections. Other types of preparations are exceptions. So I think that it will be easier for you to learn exceptions. For example, whole mount and total preparations and smear. These are very rare for us. Almost all the histological preparations that we will learn, they are sections, actually. And all of those that are in your album. Your picture number 1 is this, the neuron surrounded with glial cells. This is section. Your picture 2 is the section of muscle fibers, longitudinal and cross-sectioned. This is also section. And number 3, the ear cut. Ear auricle with cartilage tissue. So this is also the sections. So all your pictures are sections. Okay, if you have any other questions.
Thank you so much, Professor.
Okay, so have a nice day. Goodbye, everyone.
Goodbye, Professor.